What if a single DNA test could help researchers discover cures, help doctors choose the right treatment, and help patients understand their future health risks, all at the same time? Assalamu alaikum. I'm Sadia Afrin from Team GenoNexus. Today, especially in Bangladesh, the gap between genomic research and everyday healthcare is too wide. Scientists analyze DNA in one system, doctors manage treatment in another, and patients are left completely in the dark. This fragmentation delays life-saving precision medicine. To solve this challenge, we present GenoNexus, an AI-powered precision medicine ecosystem that connects researchers, doctors, and patients within one intelligence platform. We offer three portals in our system. First, researchers. This portal provides scientists with a powerful command center featuring AI-driven DNA analysis, 3D genome visualization, and real-time global collaboration. Second, Care Coordinator. It gives doctor a streamlined interface to seamlessly ingest patient genomic files and coordinate specialist referrals. Finally, Patient Portal. This empowers everyday users with a secure, intuitive dashboard to easily understand their personal genetic health insights and track their results. So, what makes GenoNexus a unique is that it doesn't serve just one user, it creates a complete ecosystem where discoveries move seamlessly from research laboratories to clinical practice and ultimately to the people who need them most. At GenoNexus, we believe the future of healthcare is not reactive, it's predictive, personalized, and accessible. Now, let's move to our researcher page. This is the main dashboard. Right at the top, our system monitors for danger. Here, an AI insight alerting us to a high risk detected recently. We can jump straight through the prediction page, and we have 4 signs for our ongoing research. These cards track the total bases aligned across all our uploads, mutation count, risk level, and AI confidence score. Moving down, we find the quick access row. This is the main navigation hub. We have one-click access to all the core tools we will look up later. Finally, at the bottom of our dashboard, we have long-term tracking tools. On left, it is cohort health trend. This chart plots the overall mutation risk score over the last 30 days or 7 days or 90 days. And on the right, the recent activity feed keeps a log of all our completed sequence analysis. Here we can see our previous work. This is our analysis history, and we can see any of this work. Now let's move to upload DNA page. Here we can upload our DNA file. We just have to drag and drop our file right into this box, but before that we can choose to keep our data private or public. Once we drop the file, the system quickly checks to make sure the format is correct. Now we scroll down and can click any of these reference buttons like COVID-19, HIV, BRCA, or Ebola virus. By clicking one of these buttons, we tell the system exactly what reference to use for the comparison. And if we select BRCA file and BRCA is used for cancer screening, so a new patient profile form will pop up. And why do we need this? Because predicting cancer isn't just about DNA. Our clinical AI needs to know the patient's age and family history to calculate a truly accurate lifetime cancer risk score. There are now, let me upload the HIV file. I have uploaded the HIV dataset file and I will click the HIV genome reference, and there are two options. I can see the mutation analysis or disease prediction, so I will go to mutation analysis. This is mutation analysis page, and in this case HIV, it brings us here. At the top right we get instant summary of the mutation found. It tells us the total number of single letter changes called SNP and breaks them down by high, medium or low risk. Now if we have to understand the scientific terms, we can click quick reference and gain knowledge about it. This is the core of the page. It lists every single mutation the AI detected, showing us exactly where it happened in the DNA, what the specific letter change was, and showing us the AI confidence level and severity risk. Finally, on the right side panel, we have advanced bioinformatics matrix and also the VCF export button. Now let's check out the chromosome heat map on the side. This feature takes us the entire genome and divides it into 64 equal blocks. It gives us a view of exactly where the mutations are hiding. This block show the risk level area also. We can click any specific mutation on the table and a detailed side panel opens up. This gives us a deep dive into the single variant. It shows us exactly where it changed, like G swapping to A. Now we can click explore in chromosome map. This is the chromosome map viewer page. On the left we have our overall sequence statistics. In the center is an interactive map of the chromosomes. Because we click through from the specific mutation, the map is currently focused on right on the exact spot, and we can see the pop-up showing the variant details on chromosome 3. But finally, if we want to see the entire picture, we just click this show overall map, and this lights up all other chromosomes, allowing us to visually explore every mutation found in the entire sequence. If I select chromosome 9, I can see the details. It identified 4 variants. Now I'm going to prediction page. This is where all our mutation data is translated into the real world health insights. At the top we get a quick summary, and on the left side we have our risk models. It screens the data against multiple conditions. For this specific sequence, it screened for 3 things: AIDS detection, antiviral resistance and immune evasion risk. Each card gives us risk percentage and how confident the AI is in that prediction, and whether the risk trend is stable or rising. If we click any of these cards like AIDS detection, a detailed explanation opens up on the right. Here we can see a clear visual of the 81% risk score. It also tells us exactly which gene are causing the risk, like this CAG gene. Finally, there is a generate prevention plan button. This allows user to create an actionable plan. Next up is the pharmacogenomics page. This looks at how your specific DNA affects the way you respond to different medications. At the top, we can get a quick summary, and below that we have a robust search and filter system. A doctor can easily type in a specific drug or can select any of these filters like cardiovascular or psychiatry. It is prescribing advice into two clear columns: favorable response and high risk. Under the favorable response, we can see drugs that are safe to prescribe. Like this drug, it is showing 91% and suggesting as 75 mg per day. If a drug was dangerous or less effective, it would be instantly flagged in the red high risk column, but it is empty because there is no risk. On the right side, we have metabolic pathway status panel. It tells us how the patient's body processes drugs on a chemical level. It uses this crucial baseline data to figure out the perfect dosage. Let's take a look at the global outbreak prediction page. While our other tools focus on individual patterns or single DNA files, this is a macro level tool. It uses machine learning to forecast disease transmission on a massive scale by combining genomic surveillance data with global mobility patterns. At the top, the system immediately presents a high-level summary, and just below the alert banner is our control panel. Here, researcher can filter the prediction data by specific region or choose pathogen or time horizon. These filters directly control the transmission velocity forecast. Chart below, the green line plots historical transmission data to show where we have been when the machine learning server is actively forecasting. The red line will seamlessly project the anticipated spread into the future. This allows public health officials and researchers to literally see what's coming and stay one step ahead of the next outbreak. We have the built-in message feature, which serves as communication hub. I can see the contact list on the left side. It clearly shows who is online or offline. If we just click on a colleague name from the list, like the main chat window opens up, we will see that their name logged in at the top, and we can say hello. Or from here, researchers can securely message each other, discuss genome comparisons and collaborate on findings directly within the platform without needing any external tools.
Now have a look at one of our unique features, which is collaboration, and at the top of it the presence header displays the active team members and our live WebSocket connection. Under this hood, this WebSocket is managed by our fast API backend broadcasting system-wide updates. In the top right, we can trigger the scientific slider. This slider or drawer aggregates critical pathogenic variants, drug-gene interaction warning, and outbreak anomalies detected dynamically by our Python genomics engine. A core design pattern for researchers here is the private versus public visibility control. When working on new datasets, researchers can keep their private data values isolated. However, when a discovery requires peer validation, they toggle the visibility to public. This immediately triggers a WebSocket update broadcasting the file to the collaboration hub, so the entire team can instantly run shared alignments and annotate variants in real time. And they can do this while uploading their own DNA files or own DNA files they are willing to work. And they can do this by toggle this button. Here is the for private dataset view, and this is for the public dataset view. And look at the hypothesis board on the left. Here, researchers formulate and test hypotheses such as targeted CRISPR knockout efficiency in lunch originoids. We can select a hypothesis to view annotation, adjust the confidence slider, or read chat logs where researchers interact with our AI Nexus copilot. This copilot uses LLM-driven synthesis to advise on sample sizes and recommend sequence depth. And I have done a pseudo hypothesis here, and anyone can do this by initializing these states. I am not doing it for our time constraint. And moving to the central live activity stream, when multiple researchers are active in our portal, they will easily visible in this site on the activity site of the GenoNexus portal. And when they each researchers are uploading each sequence file for their own research purpose, it will easily logged in the live activity stream. If any researchers can upload their private dataset, it won't be visible here. Only the public datasets are visible here. And on the right, the pipeline engine displays active bioinformatic pipelines. When the genomics engine processes raw FASTA or VCF files, it runs Needleman-Wunsch alignment and variant calling algorithm like BWA, MEM2 and GATK. The engine then pushes live computational logs and stage-by-stage progress update directly to this dashboard. And if we click the launch pipeline here, and we will be able to view the launch pipeline of our latest uploading DNA files, and I have done few here. And if we click the view logs, we will view some frequent steps, and let's discuss something about it. For the industry purpose, when we upload the actual DNA file of a patient or of a person, the DNA file might take 2 or 3 days to properly visualize it, and it doesn't make sense. Like for a single DNA file, it take 2 or 3 days to visualize. So we have proposed a genomic engine or pipeline engine which will certainly scale the DNA file to such a portion where the major mutations are occur in our DNA, and they will do it by these steps like quality control, sequence alignment, variant calling, and we will be able to it by the 3D visualization. Now the 3D visualization of this and the actual visualization is same as we have done our project on the scale DNA, not the actual DNA. And now let's move to the second part of our project, which is the care coordinator and the patient portal. And in the care coordinator, we in our project we don't give the permission to the patient to upload their own DNA files. The care coordinator manages raw ingestion. When a patient submits a DNA file request, like Mr. Rahman want to analyzes his DNA over Ebola virus genomic alignment, and he has booked a request, and it will popped up in a bridge type of system which bridges our GenoNexus portal versus patient. And there are some few offline steps, like the patient might need go to the clinic and give his blood, then the DNA will be extracted from the blood, and the DNA file will come to the care coordinator portal. And this is the care coordinator portal, and our care coordinator can upload the DNA files over our client's request or patient request. Like Mr. Rahman has requested for Ebola virus, and now let's go check. Now let's assume this is the Ebola file. This is the DNA file of Mr. Rahman. And if we click open and upload the analysis and show the patient's result, it might take some minutes to view the proper visualization of the DNA. And this is the overall condition of its DNA, like the risk of Ebola, antiviral resistance, and immune evasion risk. And they can also test and lab suggestion by this copilot calling. We have used Gemini Copilot API, and something will be popped out. Let's acknowledge it. And they can also export clinical report based on this data, and also can refer the patient to this specialist. Like I want to refer Mr. Rahman to Dr. Tariq Alam. And overall report of a patient, all activities we have done will be generated through this portal. And now let's go to the Mr. Rahman portal, and the very recent DNA analysis will be popped up here. Like we have done the Ebola, and previously I have done BRCA or the ovary cancer or breast cancer analysis, and they will be able to book more tests to actual detection of the disease. Like I want to book the CBC and the lipid profile, all will be visible here. And they can also generate a total bill and download the PDF. And the patients I have referred, the care coordinator referred to the patient, the specialist, the care coordinator referred to the patients will be visible here. They can also check the others option on all, can also take the referred specialist by the care coordinator. Like I want to take the specialist given by my care coordinator, book appointment, I want to take it to the 11, confirm, and our schedule is booked. And now let's go to the GitHub. We have contributed equally on the GitHub. Behind GenoNexus lies a robust production-ready codebase managed by entirely under Git version control. Our development workflow follows a strict branch and merge strategy, ensuring that every genomic algorithm, API route, and UI component undergoes modular integration. And this is the overall project time span of our project, and this is the contribution graph. We are a group of member 3, and this is our contribution. We can also check the pulse here. Overall pull request done in 1 month. Everything will be visible here. And now let's go for the testing. We have used the Node.js test library, like Jest and React testing library here for our unit testing to guarantee reliability. And I have initiated the commands earlier. This is for the front end. We have done the unit testing and everything is test cases are passed here. And for the back end, we have used the Python library, and this is for the genomic engine. This test case also is passed, and this is another microservice or epidemiology, and these test cases are also passed. And this is all about our project. Thank you.